Hello everyone. Today we are going to discuss about thyroid gland histology and thyroid hormone synthesis. The learning objectives. At the end of this class, you will be able to describe the histological features of thyroid gland and discuss the steps of thyroid hormone synthesis. This micrograph representing the histology of thyroid gland. As you can see, the thyroid gland consists of different sizes of thyroid follicles. Each of these follicles consists of a single layer of epithelial cells called the thyroid follicular cells. And between these cells is the colloid, whereby the formation of thyroid hormone occurs in this part. Distributed among these follicles, which is not very clearly seen in this picture, are the parafollicular cells or C cells, which secrete calcitonin. Thus, the thyroid gland produces three types of hormones, that is T3, T4, and calcitonin. T3 and T4 are considered the thyroid hormones. The synthesis of thyroid hormones, T3 and T4, occurs within the colloid, and it's required two compounds, that is the iodide and the large protein called thyroglobulin. This diagram representing the synthesis of thyroid hormone, and this is one of the cells of thyroid follicular cells, and this is the inner part which is called the colloid. The first step required for the synthesis of thyroid hormone is the uptake of iodide from the blood into the thyroid cells, which is mediated by the sodium iodide symporter transporter. These transporters are located within the basolateral membrane of the thyroid cells. The transporters uses the sodium ion concentration gradient to enable the entry of iodide against its concentration gradient. Then the iodide will be secreted into the colloid by the action of sodium-independent iodide transporters, which is also called penedrin. The second step involved in the thyroid hormone formation is the synthesis of a large protein within the thyroid cells. This protein is called thyroglobulin. Thyroglobulin is rich in an amino acid which is tyrosine. Thyroglobulin is also will be secreted into the colloid. The third step is the incorporating of the iodide to the tyrosine residues within the thyroglobulin molecule that occurs within the colloid. This action is driven by the enzymes called thyroperoxidase, which is located at the luminal surface of the thyroid cells. As indicated in this diagram, adding of iodide to the tyrosine residues will result in the formation of monoiodotyrosine and di-iodotyrosine. This iodinated tyrosine then will combine in pairs to form the thyroid hormone T3 and T4. If the pair consists of one mono and one di-iodotyrosine, that will result in the formation of T3, also called tri-iodothyronine. However, if the pair consists of two di-iodotyrosine, that will form T4, also called tetra-iodothyronine or thyroxine. In the step number 4, a part of the colloid will be taken by the endocytosis process back into the thyroid cells to form vesicles. In step number 5, fusion of the vesicles with the lysosomal enzymes and breakdown of the thyroglobulin to release the pair of the iodinated tyrosine. In the last step, step number 6, the release of T3 and T4 into the blood, and the remaining part of the thyroglobulin will be recycled to form further colloid. Take note that all the steps involved in the thyroid hormone synthesis are stimulated by the TSH hormone or thyroid stimulating hormone that is secreted from the anterior pituitary gland. TSH stimulates the uptake of iodide, formation of thyroglobulin, and iodination of tyrosine and the endocytosis process. Some possible abnormalities that may occur within the thyroid hormone formation is either there is a defect in the thyroid stimulating hormone, or there is deficiency in the dietary intake of iodide, or there are some drugs that inhibit sodium iodide symporter, or drugs that inhibit thyroid peroxidase enzyme. To summarize, we have discussed about the histological features of thyroid gland, and we have learned that thyroid gland is composed of various sizes of thyroid follicles, and that thyroid follicles consist of two components, that is the thyroid follicular cells and the colloid. Then we have discussed about the steps involved in the synthesis of thyroid hormone T3 and T4, which is uptake of iodide within the thyroid cells, formation of thyroglobulin, then adding the iodide to the tyrosine residues within the thyroglobulin, and the formation of monoiodotyrosine, di-iodotyrosine, tri-iodothyronine, and tetra-iodothyronine, or also called thyroxine. Then the next step is endocytosis of the part of the colloid and formation of the vesicles. Then degradation of the thyroglobulin molecule by the lysosomal enzymes. And the last step is the release of T3 and T4 to the circulation. Thank you very much, and I hope this one is useful to you.